Okay. We're kind of jumping in to write things now with the recording, but at least it's recording now. Thank you, Edward. Okay. So, are there any things people want me to help them with, or like need guidance on, or should I just start with some of the coding? All right, so one of the ways, like if you've done some coding before, like you probably already have conda installed. If not, a simple way for it to get this work today is using like wget, and we'll sort from the source, install the program from the source. So what I'm going to do here, I'm just going to copy that command. I am going to actually, yeah, I'm going to do this. Now I should do it this other way. All right, so let me back up a couple steps. So hopefully if you're able to have access to the files that are shared, one of the folders is called adapter removal. And in that folder, you'll see that I have one sample. We have the forward read and the reverse read. So I didn't really go into this, but when reading on a machine, Illumina machine, you have the machine reading the sequencing read in like one direction from the 5' to 3', and then you have a machine also reading from the 3' to 5'. And so that's why we have, so it's like R1 is corresponding to the forward read, R2 to the reverse read. So these are not two samples. This is the same sample, but just the forward and reverse read. So I'm going to just copy that code that I have there. Here, it's going to go to the side. It's going to basically just download the thing. So very common command in terminal is called ls. So ls is just going to show me the files that are in my terminal. So if people need help with getting access, like navigating this to their where they have the file stored on the computer, please let me know. We'll take some time, but I'll try to get through like going through this tool from the beginning to end for as of right now. So as you can see, I used to only have 2 files. Now I have 3 files because I downloaded package. Let me put this in the chat. Command. So if I go back to my, okay, here we go. Yeah, there you go. I'm going to copy the next command. So each line here is corresponding to a command. Go back to the terminal. I'm going to basically untar or open up this zip file. Basically, it's just like unzipping the entire file. So now when I do ls, now I have 4 files because this one was a gzip file. So what we can think is gzip, all the things are compressed, which helps save space on your computer, like, and that, you know, is useful when you're not really needing access to it, but you need access to it, you really do need to unzip it so then you can access everything you need. So what I'm going to do next, if I can navigate, I'm going to go use what's called the cd command. CD stands for change directories. So let's put that, copy and paste that there. But now when I do ls, type right, I see all these different files, right? Because now I'm inside. So what you can think of this process that we're doing here is basically the same as if you were navigating on your computer with through different folders. So like if I brought this over here, right, I can go here. I can click on my adapter removal folder. And so that clicking on that folder there is the same thing that what I'm doing with the CD command. So then I click on that there, right? I can see all the same files in here that are listed in my terminal. So I think there's one more command. So what last command is make, and this command is going to allow me to actually run adapter removal on my system.
Sterling, apologies for interrupting. So
no,
before running any adapter removal, I was thinking maybe it would be nice to see the content of the FASTQ file. So just slash or cat the file.
Yeah, just to see like what my FASTQ file looks like.
We've been talking about the structure of FASTQ file. Right?
Yep, we can do that as well. So yeah, this will be a good time for that. It looks like because this is kind of it's running this command. This may take a little bit. So yeah, just to rehash, so we have this file structure here. But let's see what it looks like in the samples that we're going to be looking at. So I'm just to do this as that command keeps running, I'm going to open up a new shell. That's not the one I can. All right, so I want to look to see like what my FASTQ file actually looks like. So just to do this, I'm going to do a command called head. Head is going to give me first 10 lines of my file. So what we have here, we have the @ sign like we mentioned before, and then we have a string here, or a line of characters. If I'm not mistaken, I'm pretty sure this corresponds to the exact machine, the Illumina machine. You could think of it as a serial number that was run. And then we have all these other identifier information, which I can't think of on the top of my head, but if you can Google like, what's on the first line of a FASTQ file, it should be able to tell you if you need that kind of information. And then what we have here is our GCs and Ts. Now I know it's, like I said, it's only supposed to be one line, but it's because my terminal is, I'm only able to like have it at such a size. It's pushing this line onto another line here, but you can think of it, this line is actually supposed to this keeps extending. And then we have the separator, and then we have all like the quality scores right there corresponding to each nucleotide and like what's the confidence score for each nucleotide. And then we have, so that's one read. So this is for one like DNA sequence read that we're able to generate. And then we have another sequencing read right here, right? And so if we're just curious, we can like actually count the number of sequencing reads we have in our file. So what I'd like to do is this command called word WC or word count, and we do -C, and we can actually count the number of lines. So this is going to give us a number of lines. No, actually, wait, that's not the lines. I think, yeah, it's WC-L. I'm sorry about that.
I think there's a question, Sterling, in the chat.
Oh, can you modify the name after @ to be like sample 1, sample 2? You can, although like, right, we already know it's all those files that belong to sample 1 and sample 2 because it's in the actual file name itself. So I generally would not recommend doing that. like actually messing with your FASTQ. So just because generally, like you try to standardize things, right? And so like if people start changing their like FASTQ files, it gets more difficult for reproducibility. And because every Luminous FASTQ file has a setup, right? And so like this, like when you're doing some coding, right? You're like, if once you got a stage, like you want to like start incorporating other data sets into your own analysis, right? And like hopefully you have like a pipeline already established, so then you can like just run the pipeline and like things could be automated. And so like and people like also have other pipelines, right? That like kind of automate, and but they're automated in a way, right, where they assume like your data is following this specific structure. And so, like for reproducibility, I would say I would like advise not like modifying the names. But just making sure, it's the key thing here is like, like we just have to know like all these DNA sequences, right? And all these things have to belong to the sample. And so as long as you have all that information just tied to this specific sample, like don't necessarily have to like change anything within that file. It's not send names. Yep. No, it's yeah. And I would say like you can't do it. I would just say like, to me, like my personal preference is not to do it that way, because then like it's again just standardized, and then it's also for reproducibility. And so what we got here, able to find out like the number of lines. So we got over 75 million lines. And so if we were to divide that by 4, that's probably not quite 20 million. It's about almost 19 million, right? Almost 19 million like DNA reads, like sequencing reads, right? And so that's a lot of data for just one sample. Again, this sample here, these forward and reverse sequences are generated using shotgun methods. All right. Is that good for the FASTQ file for you, or?
Yeah, I actually have another question. So what's the length of the reads? Because it seems to be a standard length, isn't it? But it shouldn't be, or they seem to be all the same length. Well, at least the first couple of them.
Yeah, so we can actually check that. Let's. So they seem to be all standardized here, right, to a certain length at this point, because so one of the steps that we did on this analysis, we use like what's called a pip and prep, and with that in the laboratory, when we're using that machine, we can actually select specific DNA fragment sizes, and so we can standardize like what lengths we're going to be getting. So
okay, because they look exactly like amplicon sequencing sequences.
Yep. Yeah, like identical lengths. Yep, and what you wouldn't really expect, right, with this sort of data. But also, this may change here in a bit once we start using adaptive removal. So if I bring up my slides. So I'm going to be running this command here. Oh wait, before I get ahead of myself, I need to copy and paste the other commands into my to the terminal, just so people can keep them as a record. So then everyone else can have those. Okay, now next slide. All right, so we'll be running this command here. I'll put that in the chat so you can access it. So what you'll see here is that I have a lot of different what we call options or flags when running this command. And so like we have the main program itself, or the main command, adapter removal, but with all these different options or parameters. We have like file one. So this parameter here is like, identify what is the forward read. File two, what identify or give the program what is the reverse read. And then had dash dash, another parameter trims, trim qualities, min quality, collapse, and all these other, so on and so forth. So if you're not really accustomed to doing bioinformatics, for generally like different programs, you'll have like different options where you can like kind of adjust like the algorithms or the settings that you want how the program to run. And so in this case, we're going to be using these options, and I kind of like give you the definitions and like tell you what each option here is doing. So the trims option, that's a typo. This is removing low quality bases. So in some of the sequencing reads, you may get like ends, and that doesn't correspond to any of the 4 nucleotides. And so like we really don't want to include those in our analysis. And so if that's the case, right, we need to like remove those from the analysis. We also have the trim qualities. So any qualities that are based on the Fred quality score. So if you can recall, like that 4th line corresponds to the quality scores. We can actually like set a certain threshold. So if the machine is not confident at a certain degree, we can actually remove those low quality bases as well. And then that's like, so that's what we're starting with the min quality. So that sets the minimum Fred quality. So if you're running like trim qualities, you need to set the min quality. We also have collapse. So we're going to forward and reverse reads, if they're identical, we're going to collapse them into a single consensus sequence. And then lastly, we can set the min length. So I set mine at 30 because generally speaking, and for ancient DNA, this is pretty, I would say conservative, because people go down all the way to 25, so they accept short fragment lengths. So there's kind of like a trade-off here. I wouldn't say there's like one optimum answer. So that's it for like ancient DNA, but this also can be applied to other sample studies as well. So as you lower the minimum length, right, your DNA, you're accepting smaller DNA fragments. So you'll retain more of your data, right? But when you set it to a smaller length, that means you're more prone to having more like mismatches, right? Because that DNA sequence, like short DNA sequences, can be mapped to more different references. But as you like extend the length or increase the length of DNA sequences, you can, when you're mapping, you're going to be like more confident that sequence belongs to whatever reference you're including. And so, but doing that, I would say, you're going to like lose some of your data, right? So all those sequences that are between like 25 and 29, right, they'll be dropped out and not included in your downstream analysis. So the main takeaway from all these like different options that we're doing here, if they all don't make sense, that's totally fine. But the main thing to get at here is that we're improving the overall quality of our sequencing data. And so this can be very critical for improving what we're going to be getting in our downstream analyses. All right, so I'm going to, I think I still have that pasted. Yeah, so I'm going to just run adapter removal. on my terminal. And this can take a second because as we looked at over there, we have like over 19 million reads, but even though like, that's a lot, like usually like a laptop, a basic laptop can finish this sort of analysis. So we've got 3 million reads. Looking back, I probably should have done this before and then talked about what each option is doing. That's okay. So I don't see any questions in the chat, but do people have any questions thus far about like what adapter removal is doing, or like why we're using this tool? If you're not at the point to really ask questions, that's also okay. But I mean, like don't be afraid. Like I'm happy to answer anything. If anything doesn't not make sense or not understand like why we're doing anything.
I have a question.
Mm-hmm.
So maybe you mentioned about it and I missed it. So aren't you supposed to specify the adapter sequence that the program is supposed to look for and
adapter
remove?
So like the barcode sequences or adapters?
Yeah.
Adapter removal actually already has, because it's like part of the Lumina system, it actually already has like kind of built in. So like so it's already doing that for me here. You can do it. You can do it that way. And like if these samples are actually, I skipped the demultiplexing part. So sometimes you also want to like do the barcode list, right, and do that demultiplexing stuff. In that case, you'll have to have that. But in this adapter removing step, you don't actually have to have the adapters. They can actually identify them for you. So everyone must be following along perfectly, not coming into any troubles. Looks like it's almost there. It's over 14 million, over 15 million. Should be just about done. I must miscount. And so I actually had this discussion with Poria about this yesterday, and be curious to know if anyone knows this answer, because with the sequencing technologies, not only like decreasing costs and more people are going to be doing like high throughput sequencing, like shotgun sequencing in the future. Like another like challenge of like running these sorts of analyses is that it's like these sequencing technologies also giving us higher output or greater output. So the files are going to be keep getting larger and larger and larger. And so with that in mind, right, it's like, like how, like, what are going to be able to do like our like individual laptops is going to be very limited. And so like when I'm running these sorts of analyses, like when I'm doing most of my bioinformatic like analyses. I'm actually running on my school's supercomputer, a high performance computing cluster, just because like these things can like take huge amounts of data and huge amounts of memory, to be more precise. And so like, like if you had like experience with amplicon sequencing and you're thinking about going to shotgun sequencing, like to me, that's one of the biggest like differences between the two like types of data sets is that, right? Whereas like amplicon sequencing, a lot of people can generally get through the process or do the analyses on their just like a basic laptop. For shotgun metagenomic analyses, you're usually going to have to require like some sort of high computing, like a high memory supercomputer to be able to perform everything that you need. And like, I'm going to be going through a couple steps, like next couple tools and stuff, where we won't be able to actually run them. I'm not actually going to run them today because they do take like they could take up 13 to 15 hours, and that's when like I even have like terabytes of data. Terabytes of memory, like access to terabytes of memory. Okay, so must, hopefully this is going to end soon. Just out of curiosity, was anybody able to get this command running? Like, can anyone like just give me a thumbs up? Or just, like in the chat to say, got it, or if not, that's okay. Let me ask everyone, this is like, does anyone need help just navigating the terminal to like get to where their files are stored on the computer? I can also help you on that. If you have access to it, I know Jorge is having trouble getting Ubuntu installed, but if you have like Ubuntu installed or access to a terminal, like if you need help just finding your files on your computer, we can also take some time and just get that set up right now. All right, finally finished. Yeah, and that took not too long. I mean, that probably took about 5 to 10 minutes for just one sample. But yeah, I mean, like if you had like 20 samples, right? Like you need more memory than what basically have access to. So in total, a process of 37 million over almost pretty much 38 million reads were processed. 80,000 reads were read per second. So the LS again. See, we have some new files here, right? So we have adaptive removable text. This is kind of like our settings file. We also have our trimmed R1 file, trimmed R2 file, and trimmed merge files. And so these are the three most important files, I would say, after running this tool. So trimmed R1 file, this looks like you're removing all the adapters. This file is like what the R1 file looks like after removing all the adapters. The trimmed R2 files are what the R2 file looks like. The reverse read looks like after removing all the adapters, and then the trimmed merge file is how you like the sequencing reads that overlap, or it will overlap from the R1 and R2. That's where we get a result from the trimmed merge file. So one way like kind of just check this and see what happened. Your trimmed file should be smaller or take up less data than your forward and reverse files, right? So if you look at just the raw FASTQ data, so like the R1 was 6.7 gigabytes. The R5C reverse one is 6.7 gigabytes. And then for the trimmed one, like the R1, it's only about 661 megabytes, and the reverse is 658. And then, but if we look at the ones that are able to like collapse, right? forward reverse, it's about 3.7 gigabytes. Now, what I recommend, like moving forward with your analyses, what you're going to want to work with is your trimmed merged FASTQ files. And so let's look at what that one looks like now. Let's see if there's any differences between what it looks like now versus earlier. Right, and so one of these differences now, right, that we didn't notice earlier, it's like now they're all the different sizes, right? And so this is because we removed all those low quality bases. And so, you know, things that like, you know, are just fillers basically, now they've been removed. And we're just stuck with our very high quality bases. You know, we still have some very long, good sequences. But, you know, this is good for what we can work with. But yeah, so like you definitely see some differences now versus when we just had the forward and reverse sequencing reads. Right. So what I would say you can like refer to these like trim merge FASTQ files. These are like, this is basically our analysis ready reads. And as like I mentioned previously, like this is what we're going to like run a lot of, we can run a lot of different analyses with this file. So a couple like a very common step like in shotgun metagenomics, but also in African sequence way, it's like taxonomic binning or taxonomic profiling. So in our lab, because we're very focused on ancient DNA, two tools that we use a lot are MALT and Megan. And so we won't be running these tools today, as I mentioned before, like these take up a very long time to process. Like they take up to like 13 and like 15 hours just to run on one sample. But like I want to give you like kind of a sense of like what each one's doing, and just so like because what they're doing is very common with other taxonomic profiles. So you have your analysis ready reads, and you need to map those reads to a taxonomic database, and then you can get like some taxonomic profile, you know, from those sequencing reads. And like these are very crucial steps in metagenomic analysis. And I would say like this is another step that a lot of different research teams use different tools. They use different settings, different parameters. So I would say it's very, you know, again, not say early stages necessarily, but it's still, I wouldn't say nowhere close to being standardized and having like a true like one way to do things, and that one way is correct. But I would say like, you know, the important thing here is to know that there are like trade-offs and pros and cons for different approaches. So this, so you know, MALT stands for Megan Alignment Tool, and Megan stands for Metagenomic Analyzer. So with MALT, we're mapping reads to a taxonomic database. So analysis ready reads comparing against a reference taxonomic database. Usually in our like the my analysis, that being mapped to entire microbial genomes, but you can also use like marker genes. The objective is to align reads to the most closely related sequences in a database. So MALT assigns taxonomic labels to map reads based on best match positions in a reference database. So it uses a lowest common ancestor algorithm to do this. It can do either, or it can do both local and semi-global alignments. We usually choose the semi-global option. And so in this option, we find the alignment that maximizes similarity between two sequences, while also allowing for gaps and insertions in sequences. And an alignment score is computed based by considering the matches, mismatches, and gap penalties. So if all this makes sense, that's okay. It's generally just understand like there's different ways and like how you score like how similar like the sequencing read in your sample is to a reference. So what we use is called the RefSeq database, and this is from NCBI. I believe, I'm pretty sure it's based in the US, but like there are like research teams that often all over the world that contribute to its repository. This database includes a collection of annotated genomic, transcript, and protein sequence records derived from data in all sorts of public sequence archives. So just give you another like an overview of Megan. Megan is also a versatile tool. It is designed to handle short reads, long reads, and assemble context for taxonomic profiling. It allows researchers to explore and interpret the taxonomic composition of complex microbial communities with high throughput sequencing data. And like MALT and Megan basically go hand in hand. And so the mapping files I used in Megan basically are in MALT basically used for Megan. And like, again, like it uses like the NCBI or RefSeq taxonomic database, and you can get like all sorts of a hierarchical taxonomic classification. So it allows us to assign sequences based on a mapping to like different taxonomic ranges, such as family, genus, and species. And again, I would say like both of these tools are designed mainly for shotgun data. It's not that you can't run your like amplicon data through these, but they're mainly designed for shotgun data. So, you know, like let's
may I ask you something,
yes.
Sterling? So they are a command line, I suppose, or they are standalone. How do these tools work? I suppose they are command based, right? The same as adapter removal, or maybe.
Yeah, it's pretty common, yeah. And so, like at least like in the ancient DNA field, like those tools are pretty, I would say, widely used and common. The one thing about these tools, right, is that they're extremely computationally like intensive and it require a lot of memory. And so it actually took us a long time to be able to get our lab to get set up with these two tools, like with MALT especially. Well, not necessarily just MALT, I guess, but it's mainly using MALT with the database that we're trying to use. And so we were trying to use a RefSeq database that includes like 48,000 microbial genomes, which was like I think it was like 52 gigabytes. No, I think it was more. It was much more than that. Maybe it was like 70 gigabytes total. And so like when you're having to load that into memory, and then also load your like 5 to 10 gigabytes of data into memory, it's going to require over a terabyte of memory to run. And so, you know, and like I wouldn't say it's very common at this point for a lot of institutions or a lot of research teams to have that kind of computational resources. So I wouldn't say like they are common because a lot of people can still use these tools with like smaller databases. Like some people can use like just a pathogen only database to like they map their like sequencing reads to their samples. But I would say like other groups have also been using like other tools like Kraken and Kraken Unique. And so instead of like using the alignment parameters like where I was going like, you know, local and semi-global, they actually use like it's a K-mer based algorithm, which is slightly different than alignment, like the BLAST alignment based algorithms. And so they're less computationally intensive and they require as much computational resources. And so I would say like, I don't know, you know, I'm not 100% sure, but because of that, it's more memory like efficient. I would say like Kraken and Kraken Unique are more commonly used than MALT and Megan. But so like, you know, like, so just be aware, like, you know, like there are other taxonomic profiles, and there's also like Metaflon, which is a Metaflon play a theory, where it's another way like to taxonomically profile your samples. And so there are definitely options that are out there. So like if you feel like you don't have like the resources to like use like a certain tool, know that there are like more efficient tools out there like that can also like help you like process your data and still able, you know, answer a question that you may have. But and like generally speaking, like all these taxonomic profiling tools give you what's called the biome table or biome format table. So the biome table is a matrix that contains the number of counts that a read was observed in a sample. It facilitates efficient handling and storage of large, sparse biological contingency tables. And many programs, like especially in R, QIIME2, all can like work with biome tables. So basically, this is what biome table looks like. This is where you can like have like O2U ID or feature, species, whatever you think. This could be a gene. And then you have your sample names at the top, like sample 1, 2, 3, 4, 5, and so on and so forth. And then like each number here corresponds to the number like DNA sequences that are able to map to this specific, what we call like, I would say feature in each sample. So like this sample here, 16 sequences mapped to this sample. Or to this feature here. And so like, yeah, I mean, I would say this is one of the most like important like file structures that we use like in microbial ecology and microbiome analyses. So, I mean, we talked about all these different steps, all these different tools that we've done. Oh, Arena has a question. Since these programs output a biome table with read counts, do these programs have any sort of compensation for different bacterial genome sizes? It's a larger amount of reads. So you have to think about this. It's not, they're kind of independent. So the genome sizes, right, bacterial genome size is sort of independent, right? And so, you're mapping your reads, like in most cases, to multiple genomes. So, and it's just, like your sequences that, like which genome does it best align to? So maybe your question, I'm not sure, but maybe your question is kind of like getting out of the question, like just by chance, are more reads going to map to larger genomes just because they're larger? I wouldn't say that's necessarily the case, because it all depends on how well your sequences map to the genomes. But your question is pretty interesting, right? Because we have to be aware, like database biases, I would say, and reference data biases. And so like this is a very common, like I would say, again, another very common obstacle in microbiome research at this point. And there's like tons of research that need to be done in database refining. So, like what makes, I would say one of the key differences between like microbiome research and let's say like any model organism or especially human research is like, is that we have to deal with like things like horizontal gene transfer, right? And so, multiple microbes may have like the same gene because of horizontal gene transfer. And so, like if your database has like one gene associated with one microbe, even though it actually originated from another microbe, right? Because of this bias, it's actually going to say it belongs to the other microbe. And so like, we have to be like, recognize that there is no like perfect database. I mean, we are working with genomes here, like, more representative than, let's say, just a single gene, but that doesn't mean that there are all sorts like biases from that. So it means, say, would there be differences in counts for, and so, yeah, I would say, again, I'm not sure if I completely understand the question. So again, like the counts here is the number, so it's the sequences there in your sample mapping to one of these features, right? So this could, let's say this feature, or this, let's say this species, or what, I guess it only shows up the class here, right? This is like class 1, this is like a different class. I know it doesn't extend all the way, so like it's kind of hard to see that. But like, this one may be like 10 gigabytes in size, this genome, this one may be like just 5 megabytes in size, right? it all depends on how the sequences in this sample though are mapping to these, you know, references. And so like just because this one's bigger than the other one doesn't mean necessarily you're going to have more map reads to the bigger one than the smaller one. Based on your sample, you may have more reads mapping to the smaller one than the bigger one, the bigger genome. And so I think like they're pretty independent, like in that sense, right? But I say that, but you know, like let's not like forget still, like, you know, like I was mentioning earlier with the horizontal gene transfer, that there are still biases in the reference database. But those biases are beyond the scope of just the size of the genome. They're more about these like the other factors. Okay, I hope that answers your question more directly that time. Does that answer more what you're hoping for, Irina? I think she's good. Great, awesome. All right, so moving forward, going to talk about like removing contaminant taxes. So like ideally, you know, like we try to do all these things, you know, like during the sample collection, try and like minimize the contamination that I'm introducing to a sample by wearing gloves and all these other things. I'm also like try and decontaminate samples once they get to the laboratory, you know, with bleach and UV. But that doesn't mean I removed all the contamination. And so like removing contaminant taxa is still very important. So contamination can come from many different sources. It could come from my hand, collector's hand. It could be coming from surrounding surfaces during sample collection. It could also come from laboratory reagents and equipment. So there's been a good number of studies that demonstrate that contamination and like the chemicals and reagents and solutions that people are using in their studies can actually be picked up. And so if people are not including controls and not sequencing their controls when they're performing their like microbiome analyses, it can actually have a huge impact on understanding the diversity and their downstream analyses. So that's why it's so critical in including like controls in your study design so that you can remove those potential contaminant, and not just remove, but also identify them and then also remove them from your samples. And so one program that's able to do this is called Decontam, which moves like contaminant taxa in your sample. So Decontam requires your data to be in the biome format, and it also requires metadata for each sample. There are two different methods. It has like a frequency method, but there's also a prevalence method in Decontam. We're going to be using the prevalence method today. And so for the prevalence method, we're going to be, it looks like the presence and absence across samples at each sequence feature. And that is how it's running a chi-square test to determine whether the taxon is contaminant or not. And so for this second exercise, hopefully you can open up R. Does everyone have R installed? If not, I'll just go ahead and put the link in the chat. Actually, would this be a good time for also like a 10-minute break for you? Just all right.
Yeah, I think we can take a 10 minutes.
Let's do a 10-minute break because I think I went through quite a bit there. So let's reconvene. Let's just reconvene at 1 o'clock. Let's do that. All right. And then if you during this time also, if you want to like, if you need help like installing R or anything else, just like you answer in the chat or yeah, ask in the chat because I may not be listening the entire time, and then I'll try to help you when I can. Resume recording. All right, so it's got a couple of yeses. That's good. There we go. All right, so we're going to get into our get set up here. So hopefully you're able to download the file, the R script file, along with the other files, and you can follow along with this if hopefully this is a little more interactive if you aren't able to get Ubuntu or able to work with the Linux environment for the last exercise. So what I have in the script is just like the script runs dcontam. For dcontam, you need a biome table, includes your biological samples. A biome table can be generated the number of tools, but ours was generated with Malt and Megan. You need a metadata table that specifies which samples are biological and which ones are controls in the mapping file, the metadata file. So just know like dcontam runs a chi-square test based on the abundance of sequences for species or features or genes, whatever kind of data set you're running. There's identified in the biological samples and compares them to the control samples. This step is important because removing contaminant taxa gives us better, like, or more accurate diversity estimates, reduces the chances of finding false positives in our exploratory analyses. Yeah, if I can, yeah. Yeah, so yeah, again, not inflating our diversity estimates. So like, and like the original paper for dcontam was by Davis and colleagues, and you can find a lot more information if you have like, I want to follow up, like just go to their GitHub page. So first things first, I'm not sure how much experience people have with R. I'm not going to like go over everything, like all the R basics. But again, like if you need some, like if you ever want help with R and getting set up with R, just I'm happy to do like something like one-on-one or maybe a separate session for that. Just let me know. So again, like I'm kind of just going to go through commands. I'm going to tell you what kind of each command is doing. And like if you do have questions, again, just ask me, but we're trying to keep things moving here. So the first thing to do is make sure we set our working directory to the place in our system where our files are located. So this is where I have my files located on my system. So I'm going to set that. That way I have access to files and can access them in R. Next thing I'm going to do is actually load all my packages I need to run this program. So I'm going to load library packages, and these are just going to, each package kind of has like commands that are necessary. If for some reason you're not able, like if you get like an error message, I would say like this is optional, but you may need to run like install BioC manager. And then like if let's for instance say like if you don't have FileSeek, like you had error when running trying to run FileSeek, you may have to then run this BioC manager ::: install. So like if you have trouble installing any of these packages or accessing them, you may have to go through these steps right here. So I'll pause here just to make sure everyone gets these packages up and loading. If people want to do like a thumbs up or thumbs down or some indication whether or not they're having trouble with this or able to get this done, just let me know. Poria, were you able to get everything installed? Oh, okay, okay. No, okay. No, I know how to fix that. That package is a little different. So what I'm going to do. I'll put this in the chat. So I'll put this link in the chat. What you want to do? Nope. You're going to come to this page here, and what you have to do is install .packages remotes. Copy paste that in your terminal, and then you'll have to run remotes ::: install github. So let me put this in the chat. Or something, you're one of the other people who mentioned that you are already installed. Were you able to have access to these packages? Hopefully that's a yes. Yeah. Corey, did that help you get your package installed? It's compiling. Great, great. So I'll go on to the next step, hopefully, as that's downloading. So the next step is actually get our data into R so that we can like have access to it. So I kind of like have this stuff here just saying like you need like what you would do in Megan if you were trying to if you still need to generate the biome table. So you would go into Megan, compare all your RMA files if you have that program installed. And from there, then you would like select species level. And so that's select all, and then that's what you would work with. But I kind of already did that stuff for you because then I already did that stuff and I exported it as a biome table. And so what we're going to do is like read this biome file into R as the JSON format. So I'm going to run this right here. So my data now is in R. So that I actually did a command called view data. Actually, I'll do it here so that maybe it's a little easier to see. If I just do that, what we'll see here, right, is that I have all my biological samples, all my samples here on the top. If I go, and this is working with my Georgian data set. And then I also have my controls here on the end. So these are going to be my controls. And so this is what I'm going to be comparing my biological samples to see if basically if any taxa within these samples are higher and then my other samples. And so then I can like start like remove those taxa from my analysis. So we also need import is called the metadata. And so for this metadata, I have my sample IDs here. I have the sample types, study, yes, country, slide, so on and so forth. That's what my metadata looks like. Then to run DKTM, you need your like basically your biome table and your metadata to be in what's called a phyloseq object, and this is just one of the programs that can do a lot of different metagenomic analysis. You can also, if you had tree information, you can also input tree information if you so wish. And so I'm going to be running this command. So this command merge file seek allows me to create file seek objects. So then if I just type file seek object here, it gives me kind of like a summary of what's included in this R object now. You can see I have a total of 801 taxa, or in this case, species. I also have 47 samples, and then the 47 samples, I have like 24 sample variables. All right, Horia, you're following along?
I couldn't keep the pace, so I gave up. I prefer just to watch your screen.
Okay. Are you sure? Is it? Are you having an error or?
Well, it's much easier to watch your screen, you know, than doing it myself.
All right. So as I mentioned in the slides, right, I'm going to be working with the prevalence method, which is basically just looking at the presence absence in our samples. We're not using the frequency method because that method requires DNA concentration values, which we do not have with this data. The main thing here is that in the metadata, we have to have like what we, in this case, going to be called sample type, as it's going to differentiate our biological from our control samples. So I'm going to be running this command called sample data is done negative. But I'm actually going to, sorry, I'm going to be creating an object here that is dot negative. That's my file seek object. So I'm going to put all my control samples in an object. So I'm just going to run that there. Then I'm going to run the is contaminant function to identify which species taxa are classified or identified in the negative control. And then I'm just going to look at my table, see like what taxa are identified as contaminants. So in my data, I have 694 false. So these 690 taxa are being identified as biological taxa. Whereas my I have 107 identified as true, and these are the, so I have 107 species that are identified as contaminants. Now it's important to recognize that, and this is not really probably important for most of you, but recognize that the default threshold in decontaminant is a 0.1. Again, that's when we're running a chi-square test. But based on our knowledge about like how ancient DNA is more susceptible to like contamination than, let's say, other sample types, a higher threshold is likely needed. So, you know, because ancient DNA data sets are going to be more contaminated than other samples, and maybe your samples are too, may need to raise the threshold, maybe let's say up to 0.5. And let's see, like, let's run it that way, and let's see like how like the two results compare. Because you know, as I'm like what I'm saying here is like when you raise the threshold, right, you'll be identifying more contaminants, right? But some of those contaminants actually may be true biological signals. And so like, you know, depending on whatever kind of sample types you're doing and like working with, you know, it's going to be probably very good if you can do some literature research to say, you know, have a general sense of like how much of your sample is contaminated or contaminant, or and how much is endogenous. Right. So I'm going to run this same program, like the same command as we're running up above, but this time I'm going to change one of the parameters and say the threshold is going to be 0.5. So now what I have, right, I have 674 biological ones, whereas in 127 species that are considered contaminants now. So if you want to view what species are being identified as contaminants, you look at this table here. Right, and so like all these here are false, false, false. So these all are my biological samples. All right, I guess it's, there you go, it's true. And then these species here, right, are being considered contaminants. So again, like if I look at this, when I use this threshold here, right, I'm identifying more contaminant taxa, but like this one here, right, I may be missing out on like on a oral taxon, like, because I mean, like in the name itself, right, it says species or taxon 332. So, you know, raising this threshold, I might be like removing more contaminant taxa, but I'm also probably going to be losing some biological. And so just, you know, this is where like, I hate to say like playing around, but like, you know, really try and validate what thresholds are most appropriate for your data set becomes, let's say, I'm going to say an art, but kind of like, you know, like how well, how is your understanding of your data set, and like, and how unique it, you know, it could be. All right. So after that, we would like to, you know, it's always kind of good to like visualize results. So like decontam also allows for this. And so basically we're going to be like kind of transforming our sample counts into using this function here. And to like create an object for the negative samples, the taxa controls. We're going to be creating an object for the calculus samples. And so now, once we have those two objects, we can make a data frame of the prevalence of that are in the positive and control samples or the negative samples. We can run that command there. So then we can run ggplot, which is very common to create figures in R. So when I look at the results here, this is how I'm going to interpret it. So I have what it's telling me here, 3 control samples, right? And then I have over 40 biological samples. So non-contaminants are going to be ones in orange or false, right? And so like these are the ones that should be higher abundance in the biological samples, but they're not identified in the controls. Whereas ones in the green or like the light green here, turquoise color, are going to be higher abundant in the controls, but less abundant in the biological samples. So like we do see some, I should mention this at the first, like each dot here represents a specific species or taxon. And so like some of these taxon, right, they, you know, they are present in one control, right, but it's not being identified as a contaminant because it's in like over 20 calculus samples, right? So it's more prevalent in the calculus samples than it is in the controls, and that's why these are considered non-contaminants, even though they are identified in the negative, like a negative control. Any questions about this graph here or this figure?
So this means there was a contamination in there, right? You just cannot, you know, you just don't know to what extent, so you just cannot remove the contaminant species, right? So as long as they were identifying the control, you know, one of the controls, right?
Right. Yep. That's exactly right. Exactly right. All right, so I'll keep moving along here. No one else has anything? All right, so we see a split in the blue and orange dots. But now, like, so we identify taxa, the contaminants. So like, we really want to remove those from our like downstream analyses, right? So how we can do that, we can use this program called prune taxa. So we're going to be using this right here. Just run this command. So that now when I look into this like filtered object now, right? I only have 674, right? So I only have those species that are identified as non-contaminants. So we can see that, like, you know, when we compare this from our original table, like we can see that the program clearly worked and we move forward. All right, so now that we have this like new R object, you know, if we want to like work in some other program, like so like Trim2, for instance, right? We want to be able to like export that table back into like a biome table or a text file. So what we can do to do that here, we run this phyloseq object to data frame command here. So if I run this now, right, you can see like, yeah, this object here has 674 entries, 48 columns. Right, so that's what I want. And then I can write that table to a text file. So I can do write table using the write table function, call my data frame, name the file. I'm going to have it tab separated. Row name is going to be false. And then I'm going to run it. So if I run it, everything works correctly. To go, it's popping up over here. I can see like with the timestamp, right? This file was created. So and it's back over here. So yep, and everything looks like a biome table. So just as like kind of a shortcut, like because you know, like looking at biome tables in a text file can be cumbersome. I usually try like open them in Excel because then it's a little easier to manipulate and manage. So yeah, so like the example in the slides, right? OTU, or we can call it species. There are all my species names. Turn left, I have all my sample names as the column headers. And then I have my counts, right, for each sample here in the middle. So, yep, everything looks good. Everything looks good. And so that's pretty much it for decontam. You know, like, and I think there are like other tools being developed out there to do like, you know, to like clean your data. But the key thing here to remember, right, is like you're not able to do this if you don't have negative controls. And so like when we're doing this, you know, like in the laboratory, we're doing like, we'll have like a empty tube at the beginning of like of a batch. And we also have an empty tube on the end of the batch, like when we're doing DNA extraction. And like we'll like we'll do all the like we'll put the same reagents that we're putting in those tubes as we are with our biological samples, and you know throughout the process, and like sequence the DNA, whatever DNA is in them at the end. And so that way we can like, you know, have this comparison and see if we're getting like any of the DNA that could be like in the reagents or just in the laboratory itself, right? Whether or not those are being introduced to like our samples. And in our case, like this can be very critical for like understanding whether or not like are we potentially just picking oral microbes up in our lab, right? I mean, people have to breathe. We're wearing masks and we're doing all these precautions. We have all this personal protection equipment and safety gear on, but that doesn't mean like it's foolproof. And so like just to make sure we validate what we're doing is correct, we have to like still perform like these analyses like decontam to ensure that we're not like we're removing any kind of potential contaminants. All right. So decontaminant it's one of the simpler tools. It's not a whole lot to get it up and running and go once you have a biome table and a metadata file. Next tool I want to kind of go over, or really like it's basically like a platform that it has like many different tools and plugins in it. It's called QIIME2. And so QIIME2 is a very powerful, extensible, and decentralized microbiome analysis package. QIIME2 can work with, it works with artifacts, or means you have to like kind of import your data. So like, which is, it's not too overly complicated. All you have to know is like, you can't just look at a biome table as in itself. You kind of have to transform a biome table into a QIIME2 object that, or a format that, so that QIIME2 knows knows how to handle it. As you'll see today, like QIIME2 has like several different plugins and like to perform like all sorts of different analyses. So like what we're going to be looking at today is going to be like beta and alpha diversity of our samples with a couple of visualizations there. So again, like if you're not able, if you don't have access to Linux, I'm sorry, but maybe not as useful for you, or maybe another boring part of the workshop. But like you can either install QIIME2 with Conda, or you can install it using like natively. So I'll install these. I'll put the links to these commands, or not the links, I guess, but just these commands in the chat, just in case someone does have access to terminal and would like to try and follow along. All right, I'm going to go ahead and put these commands in the chat, just if I know not many people have access to Linux, but just so if you want to copy and paste them into your own, like a text editor or file somewhere, so then you can hopefully run them on your own at some point. I'm going to just copy them, paste them over here. But I'll go over step by step and we'll go from there. All of this should also be in the shell script that's already on, or was already included. But just going to have it here, just in case. Just going to add it in the text just in case. So I'm going to go back to Linux. All right. I am going to go into QIIME2 folder or directory. All right, so what you'll see in this directory is that I already have a lot of the files already generated. But just so that we can be on the same page, I'm going to create another directory. And what I'm going to do, I'm going to copy my files into this new directory, so then we can do it together. So we can start from scratch. All right, so all right, so we have a text file, we have a biome table. I'm going to delete the biome table because again, I really want to start from scratch. Because this is text file is what we were able to generate from the decontam analysis. And so I really want to just get us on the same page. So I have everything here in the shell script, so everything is like, you could run all of these commands at once, but what is the fun in that? I want to like go step by step, just so we're on the same page. So the first command we're going to run is going to be this one. So again, in order to get things into QIIME2, we have to have it in the QIIME format. So for QIIME2, before we get there, you have to be able to like get it in the back into the biome format. Oh, shoot, and I forgot stuff. So I also got to make sure my environment is activated. So we're going to do. Hopefully, at some point, you'll be able to get the QIIME2 like environment installed. So once you get that installed, you can do conda activate QIIME2. And take a little bit. Okay. So now what you'll see, right? Instead of base environment, now I have the QIIME2 conda environment installed. So what this does now, I can actually, I have my commands. I can run my QIIME2 commands, which is biome convert is one of them. So I'm going to run this first. Sorry, I forgot I changed the names on the script. And so I'm just going to delete that one. I try and make things simple, but then I make things too simple, and then I forget what I'm done. Okay. So now, I'm going to biome convert. So my input file is going to be data.text. My output file is going to be data.biome. So I'm converting my text file into a biome table. One of the parameters or options for this is going to be table type. So I'm going to tell it's a OTU table. And I'm going to tell I want to make it into the HDF5 format. So that's a format that's easier to access in QIIME2 to convert into a QIIME2 file. So now if I did ls, now I see I have a biome table. What's important here, you may be thinking like, what's the actual difference? So if I did a head on the text file, right, I'm able to see stuff here. So a text file is like what we call a human readable format, so it makes sense. But then if I try looking at what's inside the biome table, right, it's a bunch of gibberish and like uninterpretable. So this is like biome tables are not human readable format. And this is because they're kind of like compressed files. And so by doing this, they take up less space. Right. And so like, again, like it's 59, I guess it not necessarily takes up less space, but they're just more compact in a way, more compressed. Okay. But basically, they have the same information in them, just how they're stored is different. All right. So now I have a biome table, but now I want to I want to import that table into the QIIME2 format. So to do that, I'm going to be running this next command. So we call QIIME command, with QIIME tools, import. So I'm telling it it's the beginning. And then for my input path or input file, I'm going to call on my biome table. Type is going to be feature table frequency. Import format is biome v210 format. This is the HDF5 format. And then the output path, I'm going to say it's going to be my species.qza because this biome table is at the species level. That's why I'm naming it the species.qza file. Uh-oh. The screen sharing stops. Does that mean it's overheated or? Or maybe because it's just, sorry, technical difficulties here. Oh, maybe just shut up. Okay. All right, sorry about that. Okay, so back at it. All right, so now I have a species.qza file. What I want to do first, I'm actually going to want to filter that table. So I still have my controls in that table, right? But I don't really need them at this point, right? Because I've already removed all the contaminant species out of it using decontam. So what I'm going to do is run QIIME feature table filter samples. And so what I'm going to do is actually call on species.qza file, then use my metadata file. And then I'm going to tell everywhere in the metadata where keep, where's the Y. So, and the column like per, so they have a column in my metadata data file called keep. And only in my biological samples I put Y, my control samples I put in. And then I'm going to tell the program to call my filter samples into like into the program. So I just realized though I did not copy my metadata file. And I also renamed it too. All right, so what I'll have to do is name this so it's like that. Keep on. I have to copy this over here. So same commands. I mean, I decided to do a little rearranging. For time, feature table, filter samples, table, species.qza, metadata file, keep, and then filter table, species filtered .qza file. So if I run that, it should work. All right, and then just kind of, I like to do like some double check something. So, right, my filter QZA file is a different size than my species.qza file, right? So, that's telling me, right, that something happened, right? Something worked. It's not completely, like it's not the exact same. All right, so one of the things in microbiome studies is that or a common thing is that you may have some like very poor quality samples, right? And this is pretty common in ancient DNA. They may just have some low quality samples. So like any samples that don't usually have like at least 1,000 counts, I would say like we usually don't want to include those in our analyses. And so I'm gonna like remove samples with less than 1,000 counts with this command here. So I'm going to do chime feature table filter samples again. In this, I'm going to say my table of species dash filtered samples, and then I'm going to use this parameter here called p-min frequency at 1,000. So any samples here that don't have at least 1,000 sample sequences are going to be removed. So I'm going to be running that. With that. So yeah, and then I guess I don't have it here, so I'll type it out. So we also have in microbiome like data, usually like some like not necessarily like I guess they can be considered low abundant taxa, right? But like taxa that only like are like we only have like let's say like 5 or 6 counts in one sample, right? More than likely, those are going to be what we call spurious mappings. And so like, it's just like, like that sample actually, like we had DNA sequences mapping to one sample, but it doesn't mean it's necessarily like a true like signal. It's more than likely a false positive. And so like when we have those like kind of samples, we really, or those species or features, we really usually want to get rid of. So when we do that, what I'm going to do is to do that, we can do chime feature table, and then filter features. And then that's not what I wanted to do. I guess my keyboard's Romanian now. That's okay. What we'll do here, so we're going to filter the features instead of the samples. And so for my input table this time, right, I'm going to call my most recent table that I just made, then do P and frequency. Now, again, you could like there's no like set threshold or like one number of what your pin mean frequency or the number of sequences like of a feature has to be like in your data set. But I usually like to be on the more conservative side, so I try to have a little higher threshold just so that my data is, like I'm only reporting like true positive signals. So what I usually do here is like 100. So like if that feature is not, we don't have at least 100 counts in the biome table, I'm just gonna get rid of it. People sometimes drop that threshold to 10. Some people drop it to like even 2. I generally I like to have at least 100. Then we have to create a new table, like call it a new name. So I'm going to do species samples 1,000, and then features 100. So you can name this whatever you want. I do this naming convention because then I know like what part of the pipeline each like file, how it was generated. So then like it's easier for me to go back when I include these kind of steps. And then I can push enter.
So Sterling, that's 100 counts, notwithstanding the number of samples, right?
Yeah, the whole biome table. Not just it doesn't have to be 100 counts of one sample. It has to be 100 counts total inside the entire data set. So like it could be like 10 in one sample, 90 in another. If it's that way, it's going to be kept. If it's only like 10 in one sample and 10 in another, so total of 20, it's going to get removed. All right, so this is good, but like again, like if I try to do like if I kind of like just want to see what's in these files, right? I'm not able to do that. So what you have to do at this point, let's like to get like a sense of like how we impact our data, let's like summarize our tables. So to do that, you chime feature table summarize. Then you put an input table, species samples table. So that's what we have there. And then we need a visualization. So Chime 2 basically has like two file formats. One is a QZA file, that's a Chime artifact. Those are not able to be visualized. But then it also has QZB files, and those files are meant to be visualized. So what I'm going to call my file, that's going to be my output file. It's going to be the same as my input file, except I add the QZB extension at the end. All right, so to view these files, generally you can do Chime tools, view, species, samples, features, and then B. But because I know like, especially if you're a beginner at this, like terminal can be difficult to navigate. So I'm going to show you how you can also like look at these files on your browser. So what I'm going to do, I'm just type Google Chime2 view. So I can click on the first link I see here. So this interface can view QZA and QZB files in your browser without uploading the server. Click here to learn more. So what I'm going to do, go back to my folder where I have all the data and navigate to the directory where I had generated today. And so what I'm going to do is just going to click on this file, drag it over here, and then I kind of get a table summary of everything. So I'm left with 42 samples in this data set. Number of features, 200. So I have 200 species at this point. So significantly drop, I would say, from the 600 that was after decontam. And then the total frequency, I have over 53 million counts. And that's for my like overall summary, but Chime2 also tells me like the frequency per sample. So I have a sample with the lowest frequency was 8,673, whereas the frequency per sample with the most had like over 3 million. And so I generate a CSV file if I want to look at per sample. And these are like very good files to like have in handy, like, especially once you get to publishing or putting your manuscript together. right? So then you have like kind of like summary of the results. So for this sample here, I had over 3 million counts left. Whereas down all the way down here, I had just 8,673. So for our next step, this number here is going to be important to know, 8,673, because our next analysis is going to require verifying our data. So I'm just going to cancel that. So Chime has a option where you can perform like what's called core metrics. And this core metrics option, like it gives you a couple of different like diversity estimates and like tables and visualizations that can be very useful. And it can be done like in a very quick manner. It's like a very like pipeline that's very efficient. So I'm going to do Chime diversity core metric. Then my input table. It's not temp. Whatever it's for. Oh, I see. I see. Okay. Yep. So typos are very bad in Linux and Unix. Okay, I understand now. So I'm going to call on my file that I generated, right? For this, I'm going to use the QZA file. And then P sampling, I'm going to go back, I'm going to use the number for the lower, like those with the sample with the lowest amount of sequencing counts. That's because I can't verify it. So what I'll go into verifying here in a second, but I can't verify any higher than what the sample with the lower lowest number. So verifying is basically just subsampling. So it's like it's a normalization technique where, you know, just by chance, you know, like you may have one sample with like millions and millions of counts and another sample with very low counts. And so you may think like, you know, it's not like an apples to apples comparison when you have so much data for one and you don't have it as much for another. So one way to kind of account for this like differences in amounts of data, if the assumption is correct, like you know, like you shouldn't, like the differences shouldn't be that great, is that you can like verify or subsample them to the same sequencing depth, or not sequencing depth, but the same count total. So that's why I'm using 8673 here. So then I'm going to call on my metadata file. And then I'm going to need to put an output directory. I'm going to just say analysis species. I'm just going to let that run. All right, so generating a whole bunch of files. This is exciting. So now if I did ls in my current directory, I see that I have a new directory over here. So I'm going to go into that. Then I'll see all the files that I was able to generate. We'll go into like details of all these different files, but we're going to go over a couple. So one of the files you see, the file names over here for these files is called break Curtis. So break Curtis is one metric to look at the beta diversity in your sample. And so it relies on relative abundance. And whereas Jacquard is unweighted Jacquard in this case, does not account for abundance, and it's just present absence of certain texting. So both are measuring beta diversity in your sample, but they're looking at them in different ways. So what I'm going to point here, though, is I want to look at this QZA file, QZV file, sorry, Bray Curtis and for QZV. So I'm going to minimize my terminal, go back here. Yes, I want to leave. And then I'm going to go into my directory, all my files. I'm going to drag it over. So if you can recall earlier in the session today, this is a PCOA plot, so a principal coordinates analysis. And what's nice about QIIME 2 is that you can kind of move around the axes here, right? And so that's nice. But what you also need to know is that each dot here again represents a sample, and then each sample is color-coded based on whatever you want. So, and like you can change the colors based on their metadata category. So I'm going to look at groups here in this example. So I'm also going to change the colors because the orange and red usually kind of, to me, bleed a little bit. So change the yellow. And so I'm able to play around with this a little bit, right? So again, samples that are close together means they have more similar microbial communities, whereas samples like far away from each other have more dissimilar communities. So I can like look at different axes. So you should know that axis 1 means this is the axis with the largest variation, and then axis 2 has the second largest variation, and axis 3 has the third. And usually you don't want to go anything beyond the three axes. And just in case you didn't know, like PCOA is like it's a reduction technique. So, you know, as we saw like with biome tables, like right, we can have like 20, 30, and you know, even hundreds of samples. We can also have like hundreds or thousands of microbial species or features, whatever, what have you. And so like when you're doing like these multivariate and like other like multiple variable like kind of analyses, right? You need some way to like kind of condense and compress that information because it becomes too overwhelming and too multidimensional, right? When you have all these different dimensions. And so really we're trying like just try and extract what are the most important dimensions or variables in our data set, like what species and stuff like account for the most variation in our data set. That's what we're really like doing here with the PCOA. Does anyone have any questions about PCOAs or anything up to this point? I know I've been kind of like flying through QIIME 2 right now.
So what's the matter with that sample up there all alone on itself? Different.
Yeah, it's very different from everything else. And to be honest, I don't know why it's like that. All I do know, and this is something that when I compare with the other side, it just has a very different microbial community. Why is that the case? I have no idea. It is like it's dating to the antique period or the Roman antique period in Georgia. And so like, is it because he just, or whoever this person was, like, did they have a different diet? Or like, you know, like, could they have been like more of like a sheep herder or something like that, whereas everyone else was like living more in the city? But then like this person was for some reason buried with the other people in the area. Or is it, I mean, is it that, or is it because like, some sort of like, other effect, like was it like a non-biological effect, like, something that happened in the laboratory that's causing this individual to have like, a very different microbiome from everyone else.
Here's another question I have. So is there any rule of thumb that you use concerning the robustness of the model? And I mean, the total amount of variation that is explained by the axis. Because so this looks pretty good, right? You have about 60%. That's a pretty, this means that 60% of the variation of the structure in your data is represented by this axis, right? But there might be other cases as well with a lower variation explained. What would be a minimum amount that you would accept as, as a pretty good model?
All right, so this is the thing with PCA, right? And so like you're talking about like modeling, like how to interpret this, right? So the main thing with PCA is that it's an ordination technique, and so like PCA, NMDS, right? So these are just trying, we're just looking for clustering here, right? But we can't really interpret now whether or not these groups are different, right? Like based on this, I gotta get back over here. I can flip this around, right? And I can look at, well, maybe the red samples are a little more over here, right? The blue samples are here, the yellow, light blue samples are more over here, right? I can flip it around here, right? I can see like, looks like there is some separation. Like there's like more separation on like maybe axis 3 and then axis 1 and axis 2. Right. But like, to say like how effective this is like as a model, right? It's hard to get into that. But this is a great question though. It's like, if I am seeing some like distinct clustering here, how can I evaluate if it's statistically significant, right? Or like if these groups are actually biologically meaningful, right? Or not maybe biologically, but at least statistically significant, right? And so that's one of the next tools that we're going to be looking into, where we run a Permanova analysis, right? And so a Permanova analysis it's independent then from a PCA, but it kind of allows us help confirm or support whether or not if we are seeing clustering based on different groups or treatments, whatever you're looking at in a PCA, whether or not those remain or if those are like actually statistically significant when you run a tool called Permanova. Does that answer your question, Horia? Like help evaluate how effective this model is?
More or less, yeah.
Okay, because it's like I guess like the key thing here is it's like by looking at a PCA, I'm never going to say, well, I mean, like I can't say we have some distinct clustering over here, right, and some distinct clustering over here, and so those microbial communities seem to be very different. But I wouldn't ever say I can never say oh, I can't say these are statistically significant, right, until I run a test like Permanova to support that under interpretation of this plot.
Shall we maybe take another break, or you want to keep on?
I'm hoping I want to try and get through Chime 2, if that's okay. And then before we run the last tool, we'll take a break there.
Sounds good.
All right, so let's go ahead and run a Permanova test. Oh, no problem. I hope you enjoyed everything. If you don't, hopefully you can maybe watch the video some point in the future. All right, so hopefully this works. So I'm going to go back to my terminal. I'm going to run what's called a Permanova test. So I'm going to do time, diversity, beta group significance. So I'm going to be using my Gray Curtis distance matrix. I'm going to be calling my metadata, and then I'm going to be calling on a group, just this group column. And I'm going to be method I'm going to be using here is called Permanova. So Permanova basically stands for permutational ANOVA. So it under like in the most basic form, it is a ANOVA test. But because microbiome is usually non-normally distributed, you have to we can run just a basic ANOVA where and like ANOVAs assume that your data is normally distributed. But to get around that, a person by the name of Marty Anderson and colleagues discovered that like Permanova is a very powerful tool for like microbial ecology data sets that are non-normally distributed, and that they can be highly effective in understanding like whether or not the statistical significance between two microbial groups are actually distinct. So I'm going to run this command. That loads. Where am I? Ah, sorry. Because my metadata is in the previous directory, so I'm going to have to run it like this. So generated here. So here are my Permanova results. So method name Permanova is same. The statistic name is pseudo-F. I have 42 samples, 3 groups. The statistic is very important, I would say here, because that's like the magnitude of difference between like different groups. And then the p-value, I'm not going to go into all the basic p-value, but basically what p-value is saying is like the groups are actually statistically significant based on my observations. Come on. And so, I have this like this, even though I wasn't seeing like necessarily, very distinct clustering, let's say in the PCA, like this test here basically like kind of supports that, there are differences between like groups 1, 2, and 3 overall. What's nice about CHIME 2 too, it's like I have all these group significance plots. That I can also run, right? And so like these are just another like visualizations you can include. Now I did forget one option, so what I'm going to do is run this command again. That should work. Ah, sorry. So I did forget. So overall, what this is telling me is like there are overall distances between groups, but if I rerun that command, now upload it again. Still statistically significant, but then I can also get pairwise comparison Permanova results. So in this case, I can see whether or not the microbial communities between groups 1 and 2 are actually different, which they are not. I can also see if the microbial communities between groups 1 and 3 are different, and they are not. So likely, most likely, like most of the differences, why I'm saying overall differences is because of the differences between groups 2 and 3. When I see statistical significance. although it's not statistically significant after doing correcting for it, multiple tests. But I still see a pretty, like, it's still, I wouldn't say large, but substantial pseudo F value.
I might say it's marginally significant, right? Sometimes people accept even values between 0.05 and 0.1.
Exactly. And it's like, like it's a lot of these times, like, I mean, microbiome, like we talked about, like all these challenges and obstacles, right? So like a lot of these times, right? So it's like with just one data set, right? It's going to be more exploratory than I would say confirmatory. Whereas like, we're just saying like we're finding general trends, but we need further investigation. We need to see like if these trends are holding true in other studies that using their own methods and if they can be like reproducible and replicated. All right, I'm gonna just try to keep going. So that's beta diversity. Well, you can do beta diversity, and one way to do beta diversity and try. We can also look at alpha diversity. Shoot. So let's try. I see now. Shoot. That's, I need it. Just, yeah, that's what I need to do. So I need to create a file that's called observed features. So just because of time, I'm going to try to go through this a little quicker, but again, hopefully if you watch the video and you still have questions, just continue. Like you can always email. All right, so we got the object created. Because we do have one more tool after this, if everyone can just hold on. So, and I do want to make sure you have a good break too before we finish up the day. The last analysis. All right, so maybe sorry. To be consistent, I'm going to again open it up in the browser. So I'm going to open up my alpha diversity results over here. So we have some nice box plots. So observed features here is the same as observed species. So like 20 here is like 20 species, 25. So what we have here at the median, we have like 42 species are identified on it, like across the group 1. Group 1 group, and group 2 has, on average has a lot more species, right? It has about 60 than the group 1. Then group 3, all right, it's going to be closer to 50. So like we see the medians across these are each different. And then like we also performed a Quest-Wallace test, see if those differences are actually significant, and they are not here. So we have an H value of 4.92. Again, you can just think of that as like the magnitude of difference, whereas p value is like what are the chances of getting these results. And we have like a 0.9, so not significant. And then we can also do like, again, pairwise comparisons. And we still get H values, P values for each pairwise comparison, along with the corrected value, a Q value. And again, nothing significant. So we see some marginal significance somewhat maybe in the beta diversity analyses. We're not seeing significance or significant differences in the alpha diversity, just to summarize those. Does anyone have any questions about alpha diversity or beta diversity? If not, let's take, I would say like, let's take like a 12-minute break and we'll reconvene for at 7:30 and then finish up the day. Yeah, and to all of you who are still here, thank you so much. This is, I'm hoping that you enjoying this, or I mean, you know, if it's very unclear, hopefully it, you know, at least just introduces you to like a lot of like, you know, like what this like microbiome research is like and some tools that are commonly used in the field. All right, let's take a break and then we'll finish up the day with one more tool and then we'll call it quits. that, like moving forward, like if you ever have questions that you come into like obstacles, like this, like it's very encouraging at least for you to conduct your own like microbiome research, and you can always like use me as a resource. So if people have questions, shoot them at me, and or if people are just too tired and they're ready to go home for the day, I'm also okay with that. I mean, it's been a long day now.
Well, thank you very much, Sterling. For me, it was very interesting. I'm pretty sure that for others as well. So, mulțumim încă o dată tuturor și for any other additional questions, or you are free to contact Sterling, I guess, over email, right?
Sounds great. Yeah, for sure.
Yeah. So, shall we call it a day?
Let's call it a day.
Ok. Mulțumim încă o dată și o zi bună.
Thank you everyone for coming out. Take care.
O zi bună să aveți. Have a good day.